keeping your naivety alive throughout the whole process is important. Even if people tell you that this cannot be done and I have seen this before and that will never work, well, it might be true, but you might, on the other hand, do it slightly differently than it's been done before. And therefore, you make it work when everyone else has told you this will never work. So I have always tried to keep this youthful naivety alive, that I don't try to listen too much to myself that this will not work because it hasn't worked before. I try to be keeping this alive, this naivety that, well, if I do it slightly different, it might work this time. And never, ever to give up. If you give up, you will definitely not succeed.
Hey there, welcome to the Smart Biotech Scientist podcast. I'm your host, David Brühlmann, and today we're diving deep into the fascinating world of cell and gene therapy with an extraordinary guest. Lars Brandén has walked the path from groundbreaking academic research at Karolinska Institute to building a revolutionary high-throughput cell biology center at Columbia and Yale. Now serving as the Director of Biology at Kolibri in Paris, Lars brings us a unique perspective on bridging the academic-industry divide. In part 1 of this special episode, we'll explore his remarkable journey and uncover the future of personalized medicine. So grab a coffee and let's dive in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. To what extent will AI enable us to reduce the risk of failure? Because still nowadays about 90% of, or even more of all the programs fail sooner or later.
That is a problem where we're still not really there. But what I see from high throughput screening and cell signaling, for example, is that there is a lot of efforts building up data on how cells react in certain given conditions and building up organelles that you can use ex vivo, etc., to build up models of how the body would react to a specific treatment. But all of these kind of information needs then to go into the AI tools that we're using, basically the equivalent of large language models, but for body-wide cell signaling and organ-based cell signaling, how organs are dealing with things on a molecular level. That needs to be better understood before we can start feeding all this information into something equivalent on a large language model. But we are getting there. We are putting this kind of data together already today, and we are starting to build these kind of models. And once we have these models, you will see a gradual shift then of what we can simulate. To what degree can we simulate a human disease and a human body, how it reacts to certain given interventions and therapies? It will all be a matter of how well we can model the organs, how well we can model how the body will react. And we do have a large variation also in the human genome if you look on the whole population. So you can't just take one model individual and say this is our model for everything, because you do have a great variation when you look at different ethnic groups, gender and ages and body compositions and things like that. So bit by bit, we will build these models that can then give us an edge on what drugs and what kind of interventions will fail in a clinical trial. And we can filter those out before we have to even go to the clinical stage, or even to animal trials for that matter also. And I expect that the failure rate go down dramatically as we're developing these tools more and refining them more and more. We will never reach 100%. That's out of the question for the very, very foreseeable future. But we might get down to 10, 15% maybe in the future of failure rate, which would be fantastic.
This would definitely change the game.
I'm not sure about the percentages here. I'm sure people will object to the percentages here. And this was just give an idea that it will be a dramatic improvement, at least. That's what I'm sure of.
Yeah, definitely. This will totally change the game and the way we can treat patients. So besides the cost and I think the complexity, what do you believe are other key challenges in translating promising lab results into clinically viable gene therapies?
I think the biggest challenge is in scaling techniques while you maintain efficacy and safety. I mean, what works in a controlled lab doesn't always translate to human biology or large-scale production. So you also have the regulatory hurdles and manufacturing cost, and that you have a consistent quality across batches, etc. And there are significant obstacles. But companies also often overlook historical data, leading that you have to reinvent the wheel over and over again, which then might push the cost up to high levels. Then you can maybe discard the project because of cost increase there. But you need to incorporate past data to a much higher degree so you can avoid having to reinvent wheel over and over again. I must say that AstraZeneca has done really great on that. They really made a huge effort to integrate all their historical data, making sure the formats were readable and accessible. And I don't know how well they have done on that, but I know that they had a serious effort in integrating all the historical data in today's R&D work. And I think that's something we should do a lot more of. But at the same time, we need to quality control whatever data we are using there so we can rely on it. But the translation, I would say, is primarily due to scaling. Scaling is a problem. When you make something work on the lab bench and make that work for thousands of patients or tens of thousands of patients, you can see that over and over again. That is a problem that is reoccurring.
And how can we solve this scaling problem?
I think it's up to education. We need to be aware of this when we are designing methods and new technologies that we do intend to work with in the clinic. We need to have that in mind throughout the whole process, the whole R&D process. You need to be steeped in the idea that this needs to be applicable also on 1,000 patients down the road. You don't need to come up with solutions necessarily, but you need to be aware of that. And if you have that level of awareness, you will maybe make different choices also when you design how your experiment should be conducted. So you already from the beginning maybe thinking about, can I automate this down the road by doing it slightly differently while still getting the same kind of results? What would be a better way of doing this if I can make sure that I can use it in 1 liter as well as in 100 microliters? Is there something I need to think about then? I think it's a matter of education to a large degree, that we need to be given the understanding already at university levels and of course later on also what it means to take a lab scale experiment all the way to industrial scale and how that will impact how you design your experiments. That needs to be taught to a much higher degree already at university. That's my own impression of things, but I think there has some merit.
Yeah, scale up is definitely an important subject and it comes up in many discussions I have had on this podcast and with other people in the industry. So it's definitely a challenge and it's definitely something we need to get a better hold onto. And I think new technologies definitely can help us with that.
I just want to add one thing there, that we should not necessarily focus too much on scaling up, but we should also consider scaling out. It might be difficult to scale up certain type of processes, but you can always come up with an idea how you can scale it out instead and just make replicas of it basically in a very smart manner. So the scaling out should also be in our minds as an alternative to scaling up. I just want to make that point.
Yeah, and scaling out oftentimes makes more sense in the cell and gene therapy space whereas in the biologic space, it's more often scale up because you can produce in large volumes. But in the cell therapy, it doesn't make sense to produce at huge volumes. Oftentimes, it's just a matter of scaling out.
Absolutely. We also have the whole space of rare diseases that are coming into the spotlight more and more now. You can see more and more clinical trials for rare diseases, and you can see the FDA is moving also on their regulations for clinical trials to accommodate more the needs for rare diseases there. And I think in the light of that, with the rare diseases, you don't necessarily need these huge batches of all the reagents necessary to accommodate the treatment of the whole global patient population of a specific rare disease. There's over 7,000 rare diseases, and I think it's over 90 or 95% of them does not have a really good therapeutic intervention. It's incredible numbers, and when you start adding up the numbers of the patients suffering from rare diseases, you really quickly come up in astonishing numbers. And if we can facilitate the development of therapeutic interventions for patients suffering from rare diseases, very frequently very young patients, I think that's something that is worthwhile to promote as much as possible, because there's a lot of effort needed there to make that happen.
Yes, absolutely. That's an area where there is still a lot of potential and a lot of people just lack a therapy. So there is a huge need for innovation. Now, speaking of innovations, what kinds of innovations, breakthroughs do you foresee in the cell and gene therapy space to make these therapies better, more accessible, more efficacious, safer?
I have been thinking about something and also published an article about that. That is using combinations of different receptor ligands to increase the selectivity of the delivery, either for synthetic gene delivery or even for reformulating existing drugs. That's something I think you see the trend of now in vivo gene therapy, especially that you have, for example, a lot of AAV serotypes here. You have about 100 different characterized versions of AAV, which has each of them has a different kind of preference for a specific type of cells or organs. And if you take that idea and then go into the synthetic gene delivery space, transfections, you can easily see that if you combine different types of receptor ligands, you can get a really high tissue type specificity or cell type specificity. And I did this as a trial to see how it worked. I took 7 or 8 different cell types, and then I took a smaller library of receptor ligands, bioactive ones that I knew would work, and I put them into different combinations, A and B, B and C, etc. And I saw if I could get them to be preferential for a specific cell type. And it was quite interesting. Just by a relatively randomly selected library of ligands or bioactive smaller molecules, peptides, I could, by combining them in different ways, get a quite high degree of cell specificity. And I can easily see that you can then implement this for transfections, where it's done or being done already now, but you can do that for transfections and get a very targeted kind of transfection vehicle that could target specific cells and specific organs. But I can see that you can reformulate existing drugs in the same manner also, with the right kind of drug formulation, with the right kind of receptor ligand combinations, that you can take existing drugs that are systemically delivered today and might have unwanted side effects or even really bad side effects, and really target them to the intended cells by using these combinations of receptor ligands. And I'm a little bit surprised that I haven't seen much more of this already so many years ago, but you just need to see for myself if this worked, and it does work, this kind of combination of receptor ligands. And I think that could be a treasure trove by reformulating existing drugs to open up new therapeutic avenues using those, and relatively cheap because they're already approved. And then as well, using the insights that you can gain from virus receptor interactions, for example, with specific cell types, and just replicate that in synthetics, basically making transfection vehicles based on these receptor ligand combinations. I think that is something that will be increasingly interesting. And then, of course, in combination with mRNA therapeutics, that's a given. mRNA will take over so many aspects of therapeutic interventions, and we have just started to scratch on the surface of what mRNA can do.
And all this will lead to safer drugs, and what I heard is also a more targeted approach, right? to reduce sound effects and treat patients with lower volumes, for instance.
Absolutely. I mean, the problem with many of the drugs today that you see is not that they might be so inefficient, but the side effects that you get is so bad that you really feel, is it worse to not take the medication or take the medication at a dose that actually has an impact on my disorder and get the really bad side effects? But if you could cut down the side effects by targeting your drug properly to the cells of the disease origin, I see that as a huge opportunity, really. Like I said, we have so many FDA-approved drugs that could be reformulated with this kind of idea around it. I know there's efforts going on already now in this direction and has been going on for some time.
Yes, absolutely. There's a lot to come. Now, in this last part of our conversation, I would like to shift gear a little bit and just tap into your extensive leadership experience. And because you're a seasoned leader, and I'm just curious, what leadership principles have you found most effective in guiding cross-functional teams through very complex biotech projects?
Yeah, there are a few key leadership principles that really stand out when you're guiding cross-functional teams through all these complexities of biotech projects. You need to have the trust, and you need to be adaptable, and you need to focus on shared goals. And I would say trust is foundational. You need to have trust between the team members and you. And I really work hard to try to create an environment where everyone feels safe to voice their ideas and concerns. You need to be able to say what you think without being cut down. This kind of openness, I think, fosters innovation also, as people are much more likely to share their ideas when they feel supported and not threatened. And adaptability is, of course, very crucial as well. We live in a very fast-paced world of biotech where projects can pivot quickly due to new findings or external factors. And I do encourage everyone to be flexible and approach challenges with this solution-oriented mindset. But at the same time, I emphasize that it's important to stay open to different perspectives and approaches. Because if you have an open mind, you're open to ideas that can lead to innovative solutions that we might not have thought about from the beginning. And finally, also, you need to maintain a clear focus on the shared goals, because that helps to keep everyone aligned and motivated also. I think that is maybe one of the more important things, to have the end goal in mind, the big picture, as we talked about before. And I really emphasize the importance for the collective mission to ensure that every team member understands how their work contributes to the overall objectives of the project. And I would say this clarity is not only fosters accountability, which is a very important thing. Accountability is super important, but it also helps the team to rally together during tough times, which is something all startups goes through, So this is, in short, a little bit fussy maybe, but leading cross-disciplinary teams is a fuzzy kind of endeavor by nature almost. But again, you have to have a certain level of basic understanding for the other disciplines that you're working with. You need to understand something about physics, you need to understand something about electronics, about chemistry, etc. Even if your main focal point might be genetics, you still have to have an understanding for other fields. So you understand the nomenclature a little bit. So you understand where they are coming from when they're talking about things. And at the same time, being able to translate your own insights in genetics and biology into words that the engineers can understand. So it's a give and take there. That's my long take on it.
And I think we live in an increasingly cross-functional, interdisciplinary world. And I think in the future, we will have to talk to all kinds of different people even more than we have done in the past. So hence my next question is, because especially as a young scientist or as a startup entrepreneur, like what advice would you give to them to make their endeavor successful, that they're able to bridge their innovation and bring it to an industry application?
The standard question would, the answer to this would be to step outside your academic comfort zone. That's the standard one. Industry requires a different mindset focused on translating discoveries into products that benefits patients, and seeking internships and collaborations early on to familiarize yourself with the regulatory process and market dynamics. That's the standard answer, which is true. But I think that keeping your naivety alive throughout the whole process is important. Even if people tell you that this cannot be done, and I have seen this before and that will never work, well, it might be true, but you might, on the other hand, do it slightly differently than it's been done before. And therefore, you make it work when everyone else has told you this will never work. So I have always tried to keep this youthful naivety alive, that I don't try to listen too much to myself that this will not work because it hasn't worked before. I try to be keeping this alive, this naivety that, well, if I do it slightly different, it might work this time. And never ever to give up. If you give up, you will definitely not succeed. So combining a slight naive approach to things with a never giving up attitude, and also being very ready to step outside of your comfort zone. All of these things, I think, is necessary if you want to bridge this gap between academic research and industry applications. And do your homework. I've seen so many examples of people that are starting new things, and you look at what's online there and you just realize that this has been done like 7 or 8 years ago and was more or less exactly what they proposed there. Why didn't you check this before you went through all the trouble of putting together a proposal? You just lost like 4 or 5 weeks at least of your life. Do your homework also. All of this combined is what you need to keep in mind.
Yeah, smart biotech scientists, keep your naivety alive. I like that. That's a great way to see that. And don't give up. Don't give up and do your homework. As we are wrapping up, Lars, what is the most important takeaway from our conversation you want our listeners to walk away with?
Well, besides to never give up and do your homework and be naive, which I think is important, I think that interdisciplinary collaboration is essential for driving innovation in the bioprocess development and personalized medicine also. Because by effectively integrating diverse expertise, whether biology, software, or engineering, we can accelerate breakthroughs that are really significantly improving patient outcomes. And embracing this mindset of collaboration and continuous learning is crucial as we navigate these complexities of this field, because it's incredibly complex. So that's what I would like to say is the takeaway.
Thank you very much, Lars, for sharing all your insights. Where can people connect with you?
Well, the absolutely easiest way to find me is on LinkedIn. And it's easy then because you can see some of my background there and what I've done and some of the publications also. So send me a message on LinkedIn and I will try to answer you as soon as I can in that case. And I'm looking forward to hearing from anyone interested to talk to me.
Yeah, smart biotech scientists, please reach out to Lars. He's available on LinkedIn. And Lars, thank you once again for sharing your expertise, your passion, and thank you for being on the show today.
Oh, thank you, David. It was my pleasure indeed. It's always interesting to have these kind of discussions. Always.
And that brings us to the end of our 2-part journey with Lars Brandén. His insights into the future of cell and gene therapy, AI integration, and leadership in biotech are invaluable. Thank you for joining us on this exploration of where science meets innovation. Before we go, are you ready to transform your biotech development process? Our fractional CTO services help you develop biologics better, faster, and at a fraction of the cost. Are you curious? Just book a free call with me today to discover how we can accelerate your path to success. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.